Hello everyone, my name is Abbas Ghaffar and I am a PhD student from the University of Calgary. We are working with the commensal organism Staphylococcus xylosus, which is present in 90% of the barns and which is normal inhabitant of the poultry environment. We are using this organism as a model in order to see the antimicrobial resistance profiling from poultry barn bioaerosols across Alberta. Antimicrobial resistance is a silent pandemic. Recently, we went through the COVID-19 and everybody knows about the pandemic. But the problem with the antimicrobial resistance is it is silently causing untreatable infections in animals, which are not only limited to animals, they are also disseminated to the humans as well. So this actually falls in the One Health umbrella. In this study, we are using XMX-CV air sampler in order to collect the air from 5 different locations within the barn, and we are using Staph API strips for the identification of the Staphylococcus xylosus. So far, we have found this pathogen from 10 out of 14 barns. And then we did check the phenotypic antimicrobial resistance profiling of this bacteria, and we find resistance against tetracycline in 5 isolates and resistance against lincomycin in 5 out of 10 isolates. Then the phenotypically resistant isolates were further confirmed with the whole genome sequencing for the presence of the respective AMR genes. So having AMR genes for tetracycline as well as the lincomycin, which were having the phenotypic resistance, we do find some genes which were silently present and they were not expressed phenotypically. And those genes were for the aminoglycosides and the phosphomycin as well as the macrolides. And then interestingly, we also find antimicrobial resistance genes against the quaternary ammonium compounds, which means the disinfectants. So having antimicrobial resistance genes against the disinfectants is quite important to notice, because at the end of the flock, farmers are using disinfectant, and if they are using the respective disinfectants, then it will not be able to actually kill or inactivate the bacteria. So it will be a problem for the respective uh, flocks as well. So here we proved whole genome sequencing as a more precise technique for the surveillance of the AMR, and we did find some silent antimicrobial resistant genes as well as antimicrobial resistant genes for the quaternary ammonium compounds. We are thankful to all the funding agencies who actually contributed for this project, and if you will be having any questions, you can contact me. Thank you.
Did you know that 2 gastrointestinal illnesses are estimated to cost $22 billion in losses in 2025? Yes, so individually or combined, necrotic enteritis and coccidiosis silently reduce broiler chickens' well-being and body weight gain by damaging the intestine and reducing nutrient absorption. Additionally, as if the farmer's economic loss wasn't enough, it is also estimated that Clostridium perfringens, the agent of necrotic enteritis, is responsible for 30% of bacterial foodborne outbreaks in the USA. Both diseases are caused by opportunistic agents. Keep it in mind because it means that, in other words, the agent needs a susceptible host in order to cause the disease. For a long time, the poultry industry used antibiotics and anticoccidial drugs to prevent birds from getting sick by the diseases. However, there is a strong global trend of reducing the usage of such drugs due to concerns with their resistance selection and human health. You may ask then, how can we win a battle if we can't use our main weapons, right? Well, do you remember I said these 2 diseases are caused by opportunistic agents? Well, in this case, we could provide the birds with the defense tools that can not only increase their defense, but also reduce the enemy numbers. And the seaweed can be one of these stews. As seaweed has many beneficial components such as antioxidants, anti-inflammatory, and antibiotic properties. Combined, these properties can improve health status and mitigate infections. Additionally, seaweed has soluble fiber, which promotes the growth of beneficial bacteria that compete with the pathogenic bacteria, reducing their numbers. The usage of seaweed has shown to be handy, as in previous experiments, it promoted growth performance, intestinal development, and enhanced immune status. For this reason, the aim of my research project is to investigate the potential gut health benefits of seaweed meal on the diet of broiler chickens with necrotic enteritis or coccidiosis.
Mystery about the avian coronavirus. Infectious bronchitis virus, also known as avian coronavirus, has become a major threat to poultry farms because it causes a severe infection in chickens known as infectious bronchitis, which results in great economic loss due to the decline in marketable egg and meat production. Despite the virus being a respiratory pathogen, it also can disseminate to other organs of chickens, including the immune organs. The primary site of infection of this virus is the upper parts of the respiratory system, including trachea and lungs, and to be more specific, it is the outer cell layer lining, which we call as epithelium. On entry of the virus into the body, the immune system recruits an array of immune cells to fight against the virus and to remove it from the body. These immune cells can recognize the virus and capture them to get rid of the infection. Of these infected cells, some cells can enter the bloodstream and eventually migrate to other organs of chickens to establish the infection. Generally, the virus gets cleared within 7 to 10 days following infection from the infected areas. However, it has been reported that the virus persists in some organs of chickens, and one such organ is an immune organ of birds known as cecal tonsils, which had the highest viral load even after 16 weeks of infection. This draws much attention as this was not the case in other immune organs, for example, spleen. Cecal tonsils are paired structures that open to the excretory orifice of chicken. Therefore, the major concern associated with the persistence of the virus in cecal tonsil is the risk of excretion of the virus and reinfection of the birds in flocks or perhaps an outbreak. Hence, my research is focused on unveiling the immunological reasons behind this interesting scenario, and I assume that this peculiar phenomenon could be due to the presence of a favorable immune environment in the cecal tonsils. I hypothesize that the so-called favorable immune environment could be due to the natural occurrence of a higher number of specialized immune cells known as regulatory T cells in the cecal tonsils. These specialized immune cells come into action once the infection subsides to protect the birds from excessive immune responses, if otherwise could lead to destruction of its own body cells. Therefore, I speculate that cecal tonsil may have these specialized immune cells in abundance, which paves the way for delay in the clearance of the virus and hence the persistence of the infection. I hope my findings will help reveal and solve the mystery behind the persistence of the virus in the cecal tonsils. Thank you.